ΔFosB in postmortem human brain tissue samples of chronic drug addicts. This is a short overview of my presentation. The introduction is about drug addiction, which is still a global affair, and I also will show this present situation in Austria. Austria, a small country in the Central Europe with a population of about 8 million people. Then I will tell you about the development of drug addiction, then chronic drug abuse, which is responsible for the induction of ΔFosB isoform transcription factors and the detection of these isoforms in human brain samples. In flash frozen samples, FFPE, then we also applied immunohistochemistry and mass spectrometry screening. Well, the introduction, undiminished illegal use of drugs still remains a global affair. It is accompanied with rampant health problems, such as blood-borne infections like HIV, hepatitis B and C. It is also responsible for increasing rates of morbidity and mortality, causing of AIDS, liver cirrhosis, and liver cancer. However, people of younger ages are concerned particularly. For example, in the year of 2009, about 150 to 270 million of people, which represents 3.3 to 6.1% of the global population, aged between 15 and 64 years, used illicit substances at least once a year, according to the World Drug Report of 2011. Out of these, 12 to 21 million people are chronic opioid addicts. This is again connected with drug-related morbidity, mortality, and delinquency. In spite of constantly improving therapy strategies, relapse rate is still 90% and beyond, again leading to a vicious circle of severe medical problems, social exclusion, and increased drug-related crime rates. Here, our small country, Austria, embedded in the center of Europe. The situation is as follows. The amount of immediate drug-related deaths according to cause of death in Austria from the years of 2002 to 2013. As you can see, the intoxications with opioids, they are first increasing, and after the year of 2006, they are slightly decreasing until now. In contrast to this, mixed intoxications with illicit drugs, or NPS, these are new psychoactive substances without opioids, those are slightly increasing over the years. Well, the development of drug addiction and dependency, there is a four-stage theory behind. The first stage is the experimentation, which is a kind of entrance into drug using. This may lead to misuse of drugs, including risky behaviors, unexplained violence, symptoms of depression and anxiety. This may lead to the abuse of drugs, which is characterized with craving, preoccupation with the drug, and symptoms of depression, irritability, and fatigue if the drug is not used. And this finally may lead or end up in drug addiction and dependency, characterized by withdrawal symptoms and compulsive use of the drug despite severe negative consequences to his or her relationships, physical and mental health, personal finances, job security, and criminal record. These pictures demonstrate the distinction of non-addicted and addicted individual. Here you can see that due to neuroplastic plasticity, which forces the memory of addiction, the inhibitory control doesn't work anymore and the drug is used. In contrast to the non-addicted individual, where the inhibitory control still controls the motivations to avoid drug addiction or the use of drug. Chronic drug abuse or addiction leads to long-lasting adaptations in specific brain regions in the reward system and leads to neuroplasticity and finally to memory of addiction. This is combined with increase of sensitization but also development of tolerance and vulnerability to relapse. The reward system of the brain, it is the mesocorticolimbic and nigrostriatal dopaminergic pathway is regulating behavior, motor activity, reward and reinforcement in the brain. The mesolimbic part consists of ventral tegmental area, the medial prefrontal cortex and the ventral striatum, and the nigrostrial part consists of substantia nigra and dorsal striatum. The induction of high levels of stable ΔFosB isoforms happens in region-specific manner following chronic but not acute exposure to a variety of psychoactive stimuli. These are drugs of abuse, different drugs, but also natural rewards, and also antipsychotic and antidepressant drugs. And these all may lead to the complex transcriptional effects which are still not fully understood and therefore remain elusive. This picture shows where things gonna happen in the reward system of the brain, consisting of the prefrontal cortex, striatum, nucleus accumbens, amygdala, ventral tegmental area, and hippocampus. Well, the FOS family transcription factors, they belong to the immediate early genes, as well as the June family transcription factors, they also belong to the immediate early genes. And out of the FOS gene family, the ΔFosB gene is the gene of our interest because the product of it is ΔFosB, consisting of 338 amino acids, and after chronic exposure to the stimuli or drug misuse, due to splicing, ΔFosB arises or is the result of it, and it lacks 101 amino acids in the C-terminus. And further, in the N-terminus, ΔFosB isoforms are phosphorylated mainly at the serine 27 site. The biochemical basis of ΔFosB's unique stability can be explained. It is generated by alternative splicing and lacks the C-terminal 101 amino acids, which are present in the whole-length ΔFosB variants. And there are two mechanisms for the ΔFosB's unique stability. The one is that ΔFosB lacks two degron domains present in the C-terminus, and they are responsible for the degradation of ΔFosB, and when they are away, the ΔFosB accumulates in the nucleus accumbens. And in addition to this, second, ΔFosB is phosphorylated by several protein kinases in its N-terminus, which leads to further stabilization but also activation. The ΔFosB and ΔFosB heterotomize with proteins of June family, for example C-June and June-D, and thereby forming the activator protein 1 transcription factor complex, which is responsible for cell proliferation, differentiation, and transformation. And once AP1 complexes are formed, a multitude of molecular pathways are initiated, and they cause genetic, molecular, and structural alterations, and many of which are directly linked with physiological and behavioral changes observed following chronic drug exposure. This picture shows you where the degron domains, they are spliced away, and therefore ΔFosB couldn't be cannot be degrade anymore, and further in the N-terminus, at the serine 27 site, due to phosphokinases, it is phosphorylated, but also inside the rather conserved DNA binding domain, there are even more phosphorylation sites predicted. This is a very conserved region, and phosphorylation sites within very conserved DNA binding regions, they are phosphorylated through 3 calcium calmodulin kinases, and also casein kinase 2 and protein kinase sites. This picture shows you that after acute exposure to drugs, the immediate early genes, they arise quite soon, but they disappear also quite soon due to degradation, because they still have these degradation domains. In contrast, the ΔFosB isoforms, they accumulate in the reward centrum because they cannot be degraded anymore due to lack of these C-terminal domains and also due to phosphorylation. Well, almost all studies on FosB and ΔFosB were performed using animal models so far. So we conducted a study on opioid addicts. Our group consisted of the typical age, around 30. Well, we have to face the rather prolonged PMI interval, and all of them had a history of quite high morphine abuse, and the gender distribution is also characteristic for drug abuse. There are more males than females affected. Well, we determined morphine concentration in several tissues, in brain tissues, medulla oblongata, cerebellum, but also in other substances, in other tissues like blood, serum, urine, and also hair if available, to get sure that this addiction is chronic. Further, we detected 6-MeM and other psychoactive substances, performing quantitative toxicological analysis using gas chromatography and mass spectrometry. Afterwards, we isolated the brain tissues, flash frozen them, and extracted protein according to Cornell, as described by Hope, but we did some modifications. Especially in the lysis buffer, we used Triton X114 instead of X100. We precipitated the proteins to purify them and to enrich them, and afterwards we applied SDS page analysis. And to be sure, to get sure for equal protein loading, we dyed the membranes with Ponko S, and before we also dyed the gels with Comosi Blue. Further, we detected ΔFosB with several antibodies, with monoclonal antibodies, but also polyclonal antibodies. And with both antibodies, we were able to detect ΔFosB isoforms. These are the polyclonal detections, and this is the monoclonal, and this is the negative control. I have to say sorry about this rather pure quality, but I lost my suitcase and I had to improvise this presentation. Afterwards, we applied immunochemistry analysis on formalin-fixed and paraffin-embedded tissue, and we used a couple of antibodies which recognize FosB and ΔFosB isoforms, but not only FosB isoforms, also anti-CREB, the cyclo-AMP response element binding protein, which is a co-activator of transcription and acting as histone acetyltransferase. And we also applied anti-BDNF antibody, and these are some samples. Here you can see the brown nuclei, the stainings, the nuclear staining of FosB and ΔFosB. This is a sample picture of a positive staining of the phosphorylated ΔFosB. Here you can see CREB staining, also a nuclear staining, and this is the negative control. Afterwards, we analyzed the proteins out of FFPE protein tissue samples. We established a protein isolation by unblocking our FFPE archive. We extracted protein, precipitated it, and purified it. And well, we are still working on this method because it's a big challenge to isolate protein out of FFPE samples, especially from postmortem samples with prolonged PMI intervals. And this is also just a wild card. We are still working on it. And the original results where we could see here this ΔFosB signal I didn't have because, again, my suitcase is not here. But here you can see positive EDNF staining, and also June-D was positive. Afterwards, we applied a mass spectrometry screening analysis, and here we find really interesting proteins which are directly or at least indirectly connected with addiction and with this ΔFosB pathway. And well, just to pick out a few important examples of these findings, the GFAP, the glial fibrillary acidic protein, it is increased during neurodegeneration and is a marker of damaged astrocytes and microglia. Another example is FBOX only, protein 2. This controls abundance and localization of specific NMDA receptors subunits in the brain and may influence synapse formation and maintenance. The HETROG protein beta 1 is also known as HETROG protein 27. It belongs to the small HETROG proteins. Critical involvement of chaperone HSPs in behavioral sensitization to morphine identifies a biological target for long-lasting adaptations with relevance to addiction. The histone H2A type 1 turnover and exchange of nucleosomal histones in the brain still remain largely unexplored. They control neuronal and glial gene expression patterns. They have an essential role in neuronal plasticity and cognition. And the increased histone turnover represent a critical and previously undocumented regulator of cell type specific transcription and plasticity in the mammalian brain. Calcium colmodoline dependent protein kinase type 2, subunit alpha, it phosphorylates not only serine 27, but also two more sites of ΔFosB, the threonine 149 and the threonine 180, as already told you. And this induces increase of activating protein 1 transcriptional activity. And finally, serum neuron-specific amylase levels may serve as marker for intoxication, and here in this case, neurotoxicity induced by drug abuse. By the summary, ΔFosB is stable even after cessation of drug administration or chronic stimulus for several weeks, months, or even much longer, maybe even lifelong. And this leads to sustained synaptic plasticity, to addiction memory, and high relapse rates. Substantial impact on regulation and expression of numerous key position genes in the brain, like glutamate receptors, they are increased, dynorphin receptors are decreasing, NF-kappa B receptors are decreasing, STK5 is increasing, and there's also a negative feedback on C-FOS. Its stability makes it detectable even in postmortem human brain tissue samples with a prolonged postmortem interval, and it represents a key factor concerning vulnerability and relapse. ΔFosB itself, or any of the numerous genes it regulates, represent potential target for development of fundamentally novel treatment strategies for drug addiction, with particular attention to more personalized therapies when thinking of the high relapse rates. ΔFosB transcription factor induction is being considered as a biomarker for evaluation and potential therapeutic interventions for addiction. However, further studies, particularly functional ones on ΔFosB and its downstream targets to shed light on the complex pathways driving addiction, are definitely needed. This is a picture of our university in Vienna. Vienna is famous for its historical buildings, so this is one of it. This is our Department of Forensic Medicine, where Professor Risser is the head of it, and those people contributed to this work. Professor Kanz, Professor Nikolaus Klopp, but also our technicians, Renate Dinkelbach and Britta Leirer. Are the pictures of my chef and Professor Kanz and Professor Klopp. Well, Fritz Altmann, he supported the mass spectrometry analysis. And finally, Uli Resch, she helped me very much with the Western blot and the immunoplotting. Then I have to acknowledge Professor Mario Herrera-Marschitz, Professor Johann Sölkner, but also Professor Tibor Hakani, Sigurd Krieger, again Renate Dinkelbach and Britta Lara. This work was funded. And finally, Professor Daniele Risser, who enabled me visiting this conference. Thank you very much.